Hello and welcome to Chapter 15, Coordination. So most animals and plants are complex organisms that are made of multi-millions of cells. And different parts of the organism will perform different functions, right? And it's important that information can pass between the different parts of the body so that their activities are coordinated. Sometimes the purpose of this information transfer is to coordinate the regulation of substances within the organism, which we just learned about, like blood glucose concentrations and homeostasis. Other times it may be to change the activity of some part of the organism in response to an external stimulus, like moving away from something that could hurt you. There are communication systems within animals that coordinate the activities of receptors and effectors. The information they receive come from the internal and external environment. So there are receptors that detect stimuli inside the body and also receptors that detect stimuli in the surrounding environment. So in animals, including mammals, there are two types of information transfer that are used to coordinate the body's activities, and these would be nerves that transmit information in the form of electrical impulses, and also chemical messengers called hormones that travel in the blood. So we learned about some aspects of homeostasis that are controlled by hormones, and those hormones are secreted by glands, and they make up the body's endocrine system. Coordination in plants also involves the use of electrical impulses for fast responses and hormones for coordinating slow responses, and we'll take a look at those at the end of this chapter. Okay, so the mammalian nervous system is made up of the brain and spinal cord, which form the central nervous system, and the cranial and spinal nerves, which form the peripheral nervous system. Now, cranial nerves are attached to the brain and spinal nerves to the spinal cord. Information is transferred in the form of nerve impulses, and they travel along nerve cells at super high speeds. Nerve cells are also called neurons, and they carry information directly to the target cells. Neurons coordinate the activities of sensory receptors, such as those in the eye, decision-making centers in the central nervous system, and effectors like muscles and glands. There are three types of neurons, each with a different function. There are sensory neurons, which transmit impulses from receptors to the central nervous system. There are intermediate neurons, also known as relay or connector neurons, and these transmit impulses from sensory to motor. And then there are motor neurons, which transmit actual impulses from the central nervous system to the effectors. Here you can see some pictures of that. So there's the sensory neuron, okay? Again, that sensory neuron is transmitting that impulse from the receptor to the central nervous system. The relay or connector neurons that will transmit those impulses from the sensory neurons to the motor neurons, so they're kind of the middleman. And then the motor neurons themselves, which transmit impulses from the central nervous system to the effectors. This shows the structure of a mammalian neuron. This is a motor neuron in particular, which transmits impulses from the brain or the spinal cord to a muscle or a gland. So the cell body of a motor neuron is within the spinal cord or the brain, and the nucleus of a neuron is always in its cell body. Sometimes you can see dark specks in a microscope in the cytoplasm, and these are small regions of rough endoplasmic reticulum that, as you know, synthesize proteins. So thin cytoplasmic processes extend from the cell body. Some of them are short, and they can have lots of branches, and these are called dendrites. A motor neuron has highly branched dendrites to give a large surface area for the endings of other neurons, and these are things that we learned in STEM. So the axon is much longer and conducts impulses over longer distances. A motor neuron with its cell body in the spinal cord might have its axon running all the way to your toes. So axons may be really long. And within the cytoplasm of an axon, there are some organelles, such as mitochondria. The ends of the branches of the axon have a large number of mitochondria, together with many vesicles containing chemicals called transmitter substances. And these vesicles are involved in passing on impulses to an effector cell, such as a muscle cell or a gland. So a sensory neuron has the same basic structure as a motor neuron, but it has one long axon with a cell body that may be near the source of the stimuli, or in a swelling of a spinal nerve known as a ganglion. Relay neurons are found entirely within the central nervous system. And they connect sensory and motor neurons. Again, they can be called the intermediate neurons or relay neurons. For most of their length, the axons of motor and sensory neurons are protected within nerves. So this picture shows a cross-section of a nerve full of neurons. And you can see that some of these are surrounded by thick dark rings, and this is myelin, which is made of specialized cells, Schwann cells, that surround the axons of some neurons. And you can see these surrounding the motor neuron. So not all axons are protected by myelin, though. You can see that some neurons without dark rings in this picture. Okay, these are unmyelated neurons, and about 2/3 of our motor and sensory neurons are this is a difficult word, unmyelinated. So myelin is made when Schwann cells wrap themselves around the axon all along its length. The Schwann cell spirals around, enclosing the axon in many layers of its cell surface membrane. And the enclosing sheath is called the myelin sheath, and it's largely of lipid together with some proteins. And the sheath affects the speed of conduction of the nerve impulse. The small uncovered areas of the axon between cells are called nodes of Ranvier, and they occur about 1 to 3 millimeters in human neurons. The nodes themselves, though, are very small, around 2 to 3 micrometers long. Okay, so a reflex arc shows how a sensory neuron, and remember that's the intermediate neuron and a motor neuron, work together to bring about a response to a stimulus. So a reflex arc is the pathway along which impulses are transmitted from a receptor to an effector without involving conscious regions of the brain. Okay, so here you can see an example of a reflex arc. Okay, there's a reflex arc in which an impulse is passed from neuron to neuron inside a spinal cord. Other reflex arcs may have no immediate neuron, and the impulse passes directly from the sensory neuron to the motor neuron. There are also reflex arcs in the brain, for example, those controlling focusing and how much light enters the eye. So within the spinal cord, the impulse will also be passed on to other neurons, which take the impulse up the cord to the brain. And this happens at the same time as impulses are traveling along the motor neuron to the effector. The effector responds to the stimulus before there is any voluntary response involving the conscious regions of the brain. Okay, this type of stimulus, again, where the effector responds to the stimulus before there is any voluntary response involving the conscious regions of the brain, is called a reflex action. It is a fast, automatic response to a stimulus. The response to each specific stimulus is always the same. Okay, reflex actions are a very useful way of responding to danger signals, like if you touch something really hot or there's an object that's flying towards you. Okay, they're often called knee-jerk reactions. Neurons transmit electrical impulses. These impulses travel very rapidly along the cell surface membrane from one end of the cell to the other and are not a flow of electrons like an electrical current. Rather, the signals are very brief changes in the distribution of electrical charge across the cell surface membrane called action potentials. And these are caused by the very rapid movement of sodium ions and potassium ions into and out of the cell. Some axons in some organisms, like squid and, earthworms, are very wide. It is impossible to insert tiny electrodes into their cytoplasm to measure the changes in electrical charge. In a resting axon, it is found that the inside of the axon always has a slightly negative charge, a negative electrical potential compared to the outside. And the difference between the potentials is called the potential difference. It's often between -60 millivolts and -70 millivolts. In other words, the electrical potential of the inside of the axon is between 60 and 70 millivolts lower than the outside. and this difference is the resting potential. The resting potential is produced and maintained by sodium-potassium pumps in the cell surface membrane. So these constantly move sodium ions out of the axon and potassium ions into the axon. The sodium-potassium pumps are membrane proteins that use energy from the hydrolysis of ATP to move both of these ions against their concentration gradients. Okay, and I'm going to say that again. The sodium-potassium pumps are membrane proteins that use energy from the hydrolysis of ATP to move both of these ions against their concentration gradients. So 3 sodium ions are removed from the axon for every 2 potassium ions brought in. The membrane has protein channels for potassium and sodium, which are open all the time. There are far more of these for potassium than there are for sodium. So some potassium diffuses back out again much faster than sodium diffuses back in. And that's, again, because there are more gates for potassium than there are sodium. And in addition, there are many large negatively charged molecules inside the cell that attract the potassium ions, which reduces the chance that they'll diffuse out. So the result of these effects is an overall excess of negative ions inside the membrane compared with the outside. The membrane is relatively impermeable to sodium ions, but there are 2 things that influence the inward movement of sodium ions during an action potential. So there's a deep concentration gradient, and also the inside of the membrane is negatively charged, which attracts positive charged ions. So this creates a double gradient, and it's known as an electrochemical gradient. Okay, so here you can see that there are more potassium than sodium channels here, creating this negative charge inside the cell compared to that positive outside the cell. So within a small, with a small addition to the to that pump, it is possible to stimulate the axon with a brief small electrical current. If the axon is stimulated in this way, the steady trace on the computer screen will suddenly change. So a rapid fleeting change in that potential difference across the membrane is called the action potential, and it's caused by changes in the permeability of the cell surface membrane to sodium ions and potassium ions, as well as the channels that are open all the time. There are channels in the cell surface membrane that allow sodium ions or potassiums to pass through, and they open and close depending on the electrical potential or voltage across that membrane, and are therefore called voltage-gated channels. So when the membrane is at its resting potential, these channels are closed. So first, the electric current used to stimulate the axon causes the opening of these voltage-gated channels in the cell surface membrane, which allow the sodium ions to pass through. And there is a much greater concentration of sodium ions outside the axon than inside, so sodium ions enter through these open channels. To begin with, though, only a few channels are open, and this changes the potential difference across the membrane, becoming less negative on the inside. And that's called depolarization, which triggers some more channels to open so that more sodium ions enter, which causes more depolarization. So if the potential difference reaches about -50 millivolts, then many more channels will open, and the inside will eventually reach a potential of +30 millivolts compared with the outside. And this is an example of positive feedback because a small depolarization leads to a greater and greater depolarization. So action potentials are only generated if the potential difference reaches a value between -60 millivolts and 50 millivolts. This value is the threshold potential. If it is less than this, then the action potential does not occur. So after about 1 millisecond, all the sodium ion voltage-gated channels close. So sodium ions stop diffusing into the axon, and at the same time, the potassium ion channels open. So potassium ions therefore can diffuse out of the axon back down their concentration gradient. And that outward movement of potassium ions removes positive charge from inside the axon to the outside again. thus returning that potential difference to the normal standard negative 70 millivolts, and that's called repolarization. In fact, the potential difference across the membrane briefly becomes even more negative than the normal resting potential. The potassium ion channels will close again. The sodium ion channels become responsive to that depolarization effect. The sodium-potassium pump continues to pump sodium ions out and potassium ions in, and this helps to maintain that distribution of sodium ions and potassium ions across that membrane so that many more action potentials can occur when necessary. So the function of a neuron is to transmit information along itself, but how do action potentials help to transmit information along a neuron? An action potential at any point in an axon's surface membrane triggers the production of an action potential in the membrane on the other side of it. This temporary depolarization of the membrane at the site of the action potential causes a local circuit to set up between the depolarized region and the resting regions on the other side of it. And these depolarize the adjoining regions and so generate action potentials in them. So in practice, this only really happens in the experimental situation when a stimulus is applied somewhere along the axon. In the body, action potentials begin at one end, and new action potentials are generated ahead and not behind. That's because the region behind it will still be recovering from that original action potential, and the sodium ion voltage gated channels are shut tight and cannot be stimulated to open. However great the stimulus, this period of recovery is the refractory period when the axon is unresponsive, and there are several consequences of there being refractory periods. So action potentials are discrete events. They do not merge into one another. There is a minimum time between action potentials occurring at any one place on a neuron. The length of the refractory period determines the maximum frequency at which impulses are transmitted along neurons. Okay, so action potentials do not change in size as they travel, and they also don't change in size according to the intensity of the stimulus, but however long an axon is, the action potential will continue to reach that peak value of plus 30 millivolts inside all the way along. So a very strong light shining in your eyes will produce action potentials of precisely the same size as a dim light. Okay, the speed at which action potentials travel does not vary according to the size of the stimulus. In any one axon, the speed of axon potential transmission is always the same. So what is different, though, about those action potentials resulting from strong and weak stimuli is their frequency. So a strong stimulus produces a rapid succession of action potentials, each one following along the axon just behind its predecessor. A weak stimulus results in fewer action potentials per second. So a strong stimulus is likely to stimulate more neurons than a weak one, whereas a weak stimulus might result in action potentials passing along just one of two neurons. A strong stimulus could produce action potentials in many more. The brain can therefore interpret the frequency of the action potentials arriving along the sensory neuron and the number of neurons carrying action potentials to get information about the strength of the stimulus. The nature of the stimulus, whether it's light, heat, touch, or so on, is deduced from the position of the sensory neuron bringing the information. If the neuron is from the retina of the eye, then the brain will interpret the information as meaning light. If for some reason a different stimuli, such as pressure, stimulates a receptor in the retina, the brain will still interpret the action potentials as light. This is why rubbing your eyes when they are shut can cause you to see patterns of light. Okay, so speed of conduction. In unmyelinated neurons, the speed of conduction is slow, being as slow as 0.5 milliseconds to the -1, so meters per second in some cases. In a myelinated human neuron, action potentials travel at speeds of up to 100 meters per second. Myelin speeds up the rate at which action potentials travel by insulating that axon membrane. So sodium and potassium ions cannot flow through the myelin sheath, so it is not possible for depolarization or action potentials to occur in parts of the axon which are surrounded by that myelin sheath. So action potentials can only occur at the nodes of Ranvier, which are the channel proteins and pump proteins are concentrated. So local circuits exist from one node to the next. Thus, action potentials can jump from one node to the next, a distance of about 1 to 3 millimeters. And this is called the saltatory conduction. So in a myelinated axon, saltatory conduction can increase the speed of transmission by up to 50 times that in an unmyelinated axon of the same diameter. Diameter also affects the speed of transmission. So thick axons transmit impulses faster than thin ones. And that's obvious, right? Their resistance is much less. So a bird pecking at an earthworm that has just emerged from its burrow acts as a stimulus to set up impulses in these giant axons, which travel very quickly along the length of its body, stimulating its muscles to contract. The rapid response may help the earthworm to escape down its burrow. In the description of the generation of an action potential, the initial stimulus might be a small electric current. In normal life, however, action potentials are generated by a wide variety of stimuli such as light, pressure, touch, sound, temperature, or chemicals. And a cell that responds to one such stimulus by initiating an action potential is called a receptor cells. Receptor cells are transducers. They convert energy in one form, like light or heat or sound, into energy of an electrical impulse, okay, in the neuron. And those receptor cells are often found in sense organs, for example. Light receptor cells are found in the eye. Sound receptors are found in the ear. Some receptors such as light in the eye or chemoreceptors in the taste bud are specialized cells which each detect a specific type of stimulus and then will influence the electrical activity of that sensory neuron. But some other ones like touch are simply the ends of the sensory neurons themselves. So the tongue is covered in many small bumps or papillae, and each papilla has many taste buds over its surface. And within each taste bud are between 50 and 100 receptor cells that are sensitive to chemicals in the stuff that we eat and drink. Each chemoreceptor is covered with receptor proteins that detect these different chemicals. And there are several types of receptor proteins. Each one gives us information about a different chemical. And so there are 5 really: sweet, sour, salt, bitter, and savory. Chemoreceptors in the taste buds that detect salt are directly influenced by sodium ions, right? And that makes sense. These ions diffuse through highly selective channel proteins in the cell surface membrane, and this leads to depolarization of that membrane. And the increase in positive charge inside the cell is the receptor potential. If there is sufficient stimulation by the sodium ions in the mouth, then that receptor potential becomes large enough to stimulate the opening of those voltage-gated calcium ion channels. Calcium ions will enter the cytoplasm and lead to exocytosis of vesicles containing neurotransmitter from the basal membrane. That neurotransmitter stimulates an action potential in the sensory neuron that transmit impulses to the taste center in the cerebral cortex of the brain. Other chemoreceptors in the taste buds use different methods, right? So cells that are sensitive to sweet have protein receptors that stimulate a G protein, which activates an enzyme to produce many molecules of cyclic AMP. Cyclic AMP acts as a second messenger, activating a cascade to amplify that signal, leading to the closure of potassium ion channels. That would also depolarize that membrane. So when receptors are stimulated, they are depolarized, right? If the stimulus is very weak, the cells are not depolarized very much, and the sensory neuron is not activated to send those impulses. But if it's stronger, then the sensory neuron is activated and transmits impulses to the central nervous system. So if the receptor potential is below a certain threshold, the stimuli only causes local depolarization of that receptor cell. And if the receptor potential is above the threshold, then the receptor cell stimulates the sensory neuron to send an impulse. But above the threshold, action potentials are initiated in the sensory neuron. So this is an example of the all or nothing law. Okay, neurons either transmit impulses from one end to the other or they do not. As we have already seen, the action potentials always have the same amplitude. As the stimuli increase in intensity, the action potentials are produced more frequently. And the action potentials do not become bigger, they have the same amplitudes, not larger ones. So threshold levels in receptors rarely stay constant all the time, right? With continued stimulation, they often increase so that it requires greater stimuli before receptors will send that impulse along the sensory neuron. So where two neurons meet, they do not quite touch. There is a tiny, tiny gap that's about 20 nanometers wide. And this gap is called the synaptic cleft. The two parts of the neurons near to the cleft, plus the cleft itself, make up a synapse. So the mechanism of synaptic transmission, so impulses can't jump across the type of synapse that we see here. Instead, molecules of a transmitter substance or a neurotransmitter are released, and this stimulates the next neuron. So this is an outline of the sequence of events that occurs. Okay, so first an action potential occurs at the cell surface membrane of the first neuron, or the presynaptic neuron. The action potential causes the release of molecules of transmitter substance into the cleft. The molecules of transmitter substance diffuse across the cleft and bind temporarily to receptors on the postsynaptic neuron. The postsynaptic neuron responds to all the impulses arriving at any one time by depolarizing. If the overall depolarization is above its threshold, then it will send that impulse. Okay. So the cytoplasm of presynaptic neuron contains vesicles of transmitter substance, and we just saw that. There are more than 40 different transmitter substances known, and many of them are found throughout the nervous system, whereas some, like dopamine and glutamic acid, occur only in the brain. So we were going to concentrate on the synapses that use the acetylcholine as the transmitter substance, and these are known as cholinergic synapses. So you'll remember that as an action potential occurs at one place on an axon, local circuits depolarize the next piece of that membrane, stimulating the opening of the sodium ion voltage gated channels. In the part of the membrane of the presynaptic neuron, there is, next to the synaptic cleft, the arrival of the action potential. Okay, and this also causes calcium ion voltage gated channels to open. Okay, so thus the action potential does not only cause sodium ions, but also calcium ions to diffuse into the cytoplasm of the presynaptic neuron. There are virtually no calcium ions in the cytoplasm, but there are many in the tissue fluid that's surrounding the synapse. So that means that there is a very steep electrochemical gradient for calcium ions. The influx of calcium ions will stimulate vesicles containing the ACH to move to the presynaptic membrane and fuse with it, emptying their contents into the synaptic cleft. So each action potential causes just a few vesicles to do this, and each vesicle contains up to 10,000 molecules of ACH. That ACH diffuses across that cleft, usually in less than 0.5 milliseconds. Okay. So the cell surface membrane of the postsynaptic neuron contains receptor proteins, and part of the receptor protein molecule has a complementary shape to part of the ACH molecule so that they can temporarily bind. This changes the shape of the protein, opening channels through which sodium ions can pass. And these sodium ions diffuse into the cytoplasm of the postsynaptic neuron and depolarize that membrane. And I want you to think, what happens when that membrane is depolarized? Okay, it generates an action potential. Okay. So if the ACH remained bound to that postsynaptic receptor, the sodium channel would remain open, and the postsynaptic neuron would be permanently depolarized. The ACH is recycled to prevent that from happening, and also to avoid wasting it. The synaptic cleft contains an enzyme, ACH, which catalyzes the acetylcholine esterase. acetylcholine esterase, which catalyzes the hydrolysis of each ACH molecule into acetate and choline. And so then that choline is taken back into the presynaptic neuron, where it's combined with acetyl coenzyme A to form ACH again. And that ACH is then transported into the presynaptic vesicles, ready for the next action potential. And the entire sequence of events, from initial arrival of the action potential to the reformation of ACH, only takes 5 to 10 microseconds. I'm sorry, milliseconds, not microseconds. Okay, so the depolarization of the postsynaptic neuron only leads to the generation of that action potential, again, if the potential difference is above the threshold for that neuron. If not, then no action potential will be created. And the chance that an action potential is generated and an impulse sent in the postsynaptic neuron is increased if more than one neuron releases ACH at the same time, or it's over a shorter period of time. So much of the research on synapses has been done at synapses between a motor neuron and a muscle, not between two neurons. The motor neuron forms a motor end plate with each muscle fiber, and the synapse is called a neuromuscular junction. So such synapses function in the same way as described above, and an action potential is produced in the muscle fiber, which will cause that muscle fiber to contract, which we'll talk about more in just a couple slides. Okay, so the role of synapses. Synapses slow down the rate of transmission of a nerve impulse that has to travel along two or more neurons. And responses to a stimulus would be much quicker if action potentials generated in a receptor traveled along an unbroken pathway from receptor to effector, rather than having to cross synapses. So what do they do? What is their point? Synapses ensure one-way transmission, so impulses can only pass in one direction at synapses. And that's because that neurotransmitter is only released on one side. There's no way that chemical transmission can occur in the opposite direction. So synapses also allow for integration of impulses. Each sensory neuron has many branches at the end of its axon that form those synapses with many relay neurons. The cell body of each motor neuron is covered with the terminations of many relay neurons. Motor neurons only transmit impulses if the net effect of the relay neurons is above that threshold. If the depolarization of the postsynaptic membrane does not reach that threshold, no impulse is sent. So one advantage of this is that impulses with low frequencies do not travel from sensory neurons to reach the brain, and that means that the brain is not overloaded with sensory information. Okay, synapses allow the interconnection of nerve pathways. So synapses allow a wider range of behavior than could be generated in the nervous system in which neurons were directly wired to each other. They do this by allowing the interconnection of many nerve pathways, and that happens in really two ways. So first, individual sensor and relay neurons have axons that branch to form synapses with many different neurons. And that means that information from one neuron can spread out throughout the body to reach many relay neurons and many effectors, as happens when we respond to dangerous situations. And then second, there are many neurons that terminate on each relay and motor neuron, as they have many dendrites to give a large surface area. So this allows one neuron to integrate the information coming from different parts of the body, something that is essential for decision making in the brain. And also, synapses are involved in memory and learning. Despite much research, little is known still about how memory operates, but there is evidence that it involves synapses. So for example, if your brain frequently receives information about two things at the same time, like the sound of a voice and the sight of that particular face, then it is thought that new synapses form in your brain that link the two, passing that information along the particular pathway from your ears and your eyes. So in the future, when you hear that voice, information flowing from your ears along that pathway automatically flows into the other pathway too, so that your brain can picture the face that goes along with that voice. So compare how you respond when talking on the phone to someone you know well and someone you have never met. Okay, so now we're going to talk about the contraction of muscles, but and in this, in the beginning, specifically striated muscle. Okay. So we just talked about how a synapse between a motor neuron and a muscle, and how the motor neuron forms those motor end plates, and that the action potential produced is the muscle causes it to contract. Okay, so the type of muscle tissue, the striated muscle tissue, makes up many muscles in the body that are attached to skeleton. So striated muscle only contracts when it's stimulated to do so by impulses that arrive via motor neurons. Muscle tissue like this is described as being neurogenic. So you've already learned about cardiac muscle in the heart as myogenic, right? It contracts and releases automatically with no need for impulses from neurons. We have also mentioned about smooth muscle, which is found throughout the body in organs. And we learned about that in the lungs with gas exchange, as well as the alimentary canal, the walls of our arteries, atrioles, and veins. Most smooth muscle only contracts when it receives impulses in motor neurons. But smooth muscle in arteries also contracts when it's stretched by the pressure of blood surging through them. And that happens without any input from the nervous system. And that type of muscle is called smooth muscle because, unlike the other two types, there is no striations. Smooth muscle does not form smooth linings of tubular structures, such as the trachea and the arteries. The lining of those is always formed by an epithelium. The structures and functions of the three types of muscle tissues can be compared here. Okay, and always in those Cambridge exams, there are lots of questions about structure and function. So you can see they're striated cardiac and smooth muscles with the type of control. So it's obviously very important that the activities of the different muscles in our bodies are coordinated, right? When a muscle contracts, it exerts a force on a part of our body, like a bone, and that can result in a particular response. But the nervous system ensures that the behavior of each muscle is coordinated with all the other muscles so that together they can bring about the desired movement without causing damage to any of the other parts of the skeletal or musculoskeletal system. And so a muscle such as like your bicep is made up of thousands of muscle fibers. And each muscle fiber is a very specialized cell with a highly organized arrangement of contractile proteins in the cytoplasm, surrounded by that cell surface membrane. Some biologists prefer not to call it a cell because it contains many nuclei. And instead they prefer the term synictum, which to describe the multinucleate muscle fiber. The parts of the fiber are known by different terms. The cell surface membrane is the sarcolemma, the cytoplasm is the sarcoplasm, and the endoplasmic reticulum is called the sarcoplasmic reticulum. The cell surface membrane has many deep infoldings into the interior of the muscle called transverse system tubules or T-tubules, and these run close to the sarcoplasmic reticulum. The membranes of the sarcoplasmic reticulum have huge numbers of protein pumps that transport calcium ions into the cistern of the SR. And the sarcoplasm contains a very large number of mitochondria, okay, often packed tightly between myofibrils. And these carry out aerobic respiration, like we know, generating the ATP that's required for muscle contraction. Really, the most striking thing about muscle fiber, though, is its stripes or striations. So these are produced by a very regular arrangement of many myofibrils in the sarcoplasm. Each one is striped in exactly the same way and is lined up precisely against the next one, so producing that pattern that you can see in these striated muscles. And this is as much as we can see using a light microscope, but with an electron microscope, it's possible to see that each myofibril is itself made up of yet even smaller components called filaments. So parallel groups of thick filaments lie between groups of thin ones. Both thick and thin are made up of protein. The thick filaments are made of mostly myosin, while the thin ones are mostly made of actin. So now we can understand why it looks like stripes. Okay, the darker parts of the stripes, the A bands, correspond to the thick filaments, and the lighter parts, the I bands, are where there are no thick filaments, only thin. So the very dark parts of the A band are produced by the overlap of thick and thin, while the lighter area within the A band, known as the H band, represents the parts where only the thick can be seen. So a line known as the Z line provides an attachment for those actin filaments, while the M line does the same for the myosin filaments. And the part of the myofibril between the Z lines is called the sarcomere. Myofibrils are cylindrical in shape, so the Z line is in fact a disc separating one sarcomere from another and is also called the Z disc. So again, guys, this is just a matter, as so many things, of really looking at these models and really studying each part of this anatomy to understand how they work together. So the thick filaments are composed of many molecules of myosin, which is a fibrous protein with a globular head. The fibrous protein helps to anchor the molecule into the thick filament. Within the thick filament, many myosin molecules all lie together in a bundle within their globular heads, all pointing away from the M line. So the main component of thin filaments, actin, is a globular protein. Many actin molecules are linked together to form a chain, and two of these chains are twisted together to form a thin filament. Also twisted around the actin chains is a fibrous protein called tropomyosin. So another protein, troponin, is attached to the actin chain at regular intervals. Muscles cause movement by contracting. The sarcomeres in each myofibril get shorter as the Z discs are pulled closer together. It's known as a sliding filament model of muscle contraction. The energy for the movement comes from ATP, molecules that are attached to the myosin heads, and each myosin head is an ATPase. So when a muscle contracts, calcium ions are released from stores in the SR, and they bind to the troponin, and this stimulates troponin molecules to change shape. So the troponin and the tropomyosin proteins move to a different position on the thin filaments, exposing parts of the actin molecules which act as binding sites for the myosin. Now, all of this functionally should sound familiar to you. This is similar to what you learned in about enzymes, right? The myosin head binds with these sites, forming cross bridges between the two types of filament. So next, the myosin heads tilt, pulling the actin filaments towards the center of the sarcomere. The heads then hydrolyze ATP molecules, which provide enough energy to force the heads to let go of the actin. Now the heads tip back to the previous positions and bind again to the exposed sites on the actin, and then the thin filaments move as a result of the previous power stroke. So now the myosin heads bind to actin further along the thin filaments, closer to the Z disc. They tilt again, pulling the actin and filaments even further along, then hydrolyze more ATP molecules so that they can let go again. And this goes on, so long as the troponin and the tropomyosin molecules are not blocking the binding sites, and so long as the muscle has a supply of ATP. So skeletal muscle contracts when it receives an impulse from a neuron. And impulse moves along the axon of a motor neuron and arrives at that presynaptic membrane. A neurotransmitter, generally acetylcholine, diffuses across the neuromuscular junction and binds to receptor proteins on the postsynaptic membrane. Okay. So you can see here in this model the events at the neuromuscular junction as well as the events in the muscle fiber. Okay. The binding of the acetylcholine stimulates the ion channels to open so that sodium ions can enter to depolarize the membrane, which we know depolarization of the membrane generates an action potential, right? So impulses pass along the sarcolemma and along the T-tubules towards the center of that muscle fiber. And the membranes of the sarcoplasmic reticulum are very close to the T-tubules. The arrival of them, of those impulses, causes that calcium ion channel to open. The ions diffuse out down a very steep gradient into the sarcoplasm surrounding the myofibrils. So the calcium ions will bond with the troponin molecules that are part of those thin filaments, and this will change the shape of the molecules, which causes the troponin and the tropomyosin to move away, expose the binding sites for the myosin heads. Then the myosin heads attach to the binding sites on the thin filaments and form cross bridges. So when there is no longer any stimulation from that motor neuron, there are no impulses conducted along the T-tubules. Released from stimulation, the calcium ions channel in the SR close, and the calcium pumps move calcium ions back into stores in the sarcoplasmic reticulum. So as calcium ions leave their binding sites on that troponin, tropomyosin moves back to cover the myosin binding sites on those thin filaments. Where there are no cross bridges between thick and thin filaments, the muscle is a relaxed state. So there's nothing to hold the filaments together. So any pulling force applied to the muscle will lengthen the sarcomeres so that they are ready to contract again. Each skeletal muscle in the body has an antagonist, a muscle that restores sarcomeres to their original lengths when it contracts. For example, the triceps is the antagonist of the biceps. Okay, so a contracting muscle uses a lot of ATP, and we saw if there wasn't enough ATP, then the muscle wouldn't be able to contract, right? The very small quantity of ATP in the muscle fibers in a resting muscle is used up really quickly once it starts to contract. So more ATP is produced by respiration, both aerobic inside the mitochondria, and when that supply can't last, by lactic fermentation. So muscles also have another source of ATP produced from a substance called creatine phosphate. They keep their stores of this substance in the sarcoplasm, and it's this immediate source of energy once they have used the small amount that they already have that will help them to move forward. So a phosphate group can quickly and easily be removed from each creatine phosphate molecule and combined with ADP to produce ATP. So really it's creatine phosphate plus ADP yields creatine plus ATP. And in the meantime, if energy is still being demanded by the muscles and there is no ATP, the creatine is converted to creatinine and excreted in urine. Okay, so moving on to hormonal communication. So the control by the nervous system is really fast, and we've seen those impulses travel quickly. However, it's also expensive in terms of energy. All of the pumping of sodium and potassium ions that are needed to maintain enough resting potentials and in protein synthesis to make all the channels and pump proteins. Plus energy is needed to maintain all the neurons and other cells in the nervous system, like the Schwann cells. So a cheaper alternative is to use hormones that are secreted in tiny quantities and dispersed around the body in the blood. And we've learned a lot about these homeostatic functions like the control of blood, glucose concentration, and the water potential of the blood. And we know these needed to be coordinated all the time, but they do not need to be coordinated in a hurry. Hormones are ideal for controlling these types of functions. So hormones like adrenaline, insulin, glucagon, and ADH are made in endocrine glands. A gland is a group of cells that produces and releases one or more substances, a process that we call secretion. Endocrine glands contain secretory cells that pass their products directly into the blood. As endocrine glands do not have ducts, they're often known as ductless glands, and hormones are small signaling molecules. So the hormones that we already learned about previously in chapter 14 are peptides or small proteins. They're usually water soluble, and they can't pass the phospholipid bilayer of cell surface membranes. These hormones bind to receptors on their target cells that in turn activate secondary messengers to transfer the signal throughout the cytoplasm. The steroid hormones, though, that we're going to talk about are lipid soluble, so they can pass through the phospholipid bilayer. And then once they cross the cell surface membrane, they can bind to receptor molecules inside the cytoplasm or the nucleus and activate processes like transcription. So now turning to hormonal control of the human menstrual cycle. So after puberty in women, the ovaries and the uterus go through a series of changes that recur approximately every 28 days. And we know this is the menstrual cycle. So this figure here shows the changes that occur during the cycle in the ovary. In the middle of the cycle, the female gamete will be released in the oviduct. If fertilization occurs while the gamete is in the oviduct, the embryo that developed needs somewhere to embed itself to continue its development. And there is a cycle of changes that occurs in the uterus so that the lining is ready to receive the embryo, allowing it to continue its development. And we learned about this in a more general sense in STEM, right? So the uterine cycle is synchronized with the ovarian cycle so that the endometrium is ready to receive the embryo at the right time. The menstrual cycle is coordinated by glycoprotein hormones that are released by the anterior pituitary gland and by the ovaries. The anterior pituitary gland secretes follicle-stimulating hormone and luteinizing hormone. These hormones control the activity of the ovaries. So during the monthly ovarian cycle, follicles develop, which secrete the steroid hormone estrogen. After the female gamete is released from the ovary at ovulation, the remains of that follicle secrete progesterone, another steroid hormone. And you can see here the changes in these hormones during the menstrual cycle. So it's considered to begin with the onset of menstruation. Menstruation usually lasts 4 to 8 days, and during that time, the anterior pituitary gland secretes LH and FSH, and their concentrations will increase over a few days. So in the ovary, one follicle becomes the dominant one. The presence of LH and FSH stimulates the secretion of estrogen from the cells that are surrounding that follicle. And the presence of estrogen in the blood has this negative feedback effect on the production of LH and FSH, so the concentration of these two hormones will decrease. The estrogen stimulates the endometrium to grow, which thickens and develops numerous blood capillaries. So when the estrogen concentration of the blood reaches a level around 2 to 4 times its level at the beginning, it stimulates a surge in the secretion of LH and to a lesser extent FSH. The surge of LH causes the dominant follicle to burst and to shed its gamete into the oviduct. So that usually happens 14 to 36 hours after the LH surge. So here the follicle then collapses to form the corpus luteum, which secretes progesterone and some estrogen. And together these two hormones maintain the lining of the uterus, making it ready to receive an embryo if fertilization happens. Progesterone also inhibits the anterior pituitary gland from secreting FSH so that no more follicles develop. So high levels of estrogen and progesterone in the second half of the cycle will inhibit the secretion of FSH and LH by that anterior pituitary gland. So that means that there is less stimulation of the corpus luteum so that it begins to degenerate, secrete less estrogen and progesterone. So as the concentration of these two hormones decrease, the endometrium is not maintained and menstruation will begin. The decrease also releases the anterior pituitary from inhibition, so FSH will then be secreted and start another cycle. Okay, so this brings us to the discussion of birth control. So birth control means taking control over when a couple has a child, and it can involve contraception, which means preventing fertilization when sexual intercourse takes place. There are also several methods of birth control that do not prevent conception, but rather prevent an embryo from implanting into the lining of the uterus. So these can be termed anti-implantation methods, and they include the use of intrauterine devices and the morning after pill. So here we look only at methods that use hormones to prevent pregnancy. At the moment, a birth control pill is available only for women, although considerable research is happening to produce a pill that men could use. The pill was developed in the 1960s, and its introduction had a really big impact on the freedom of women to have sexual intercourse without running the risk of becoming pregnant. The pill contains steroid hormones that suppress ovulation, so usually a synthetic hormone rather than natural ones are used, but they are not broken down as rapidly in the body and therefore act longer. Some forms of the pill contain progesterone only, but most contain both progesterone and estrogen and are known as combined oral contraceptives. So there are many different types with slightly different ratios because women are not all alike in the way that their bodies respond to the pill. But with most types of oral contraceptive, the woman takes one pill daily for 21 days and then stops for 7 days, during which time menstruation occurs. For some types, she continues to take a different colored inactive pill for these 7 days. So both estrogen and progesterone suppress the secretion of, as we saw, of FSH and LH from the pituitary gland. So this is an example of negative feedback. Taking the pill daily, starting at the end of menstruation, keeps estrogen and progesterone concentrations high. And then, of course, this suppresses the secretion of FSH and LH, and that prevents their concentrations from reaching the levels that would stimulate ovulation. So this essentially mimics the natural situation during the second half of the menstrual cycle, right? It's extending it through your whole cycle. Stopping taking the pill after 21 days allows the concentrations of estrogen and progesterone to fall to the point at which the uterine lining is no longer maintained, menstruation occurs, and this reassures the woman taking the pill that she is not pregnant. So this combined oral contraceptive is very effective at preventing conception, but the women taking it have to be careful not to miss even a single day's pill, right? Because that could lead to fertilization. The combination of estrogen and progesterone can also be given by means of a skin patch or an injection or an implant. And obviously these methods will do the same thing. No menstruation will take place because you can't stop getting it. You can't stop the patch or the injection. Pills containing only progesterone may allow ovulation to occur. They seem to work as contraceptives by reducing the ability of sperm to fertilize the egg and by making the mucus secreted by the cervix more viscous and so less easily penetrated. So another one, which is a different kind, is the morning after pill. So this form of birth control is intended to be taken after a woman has had unprotected sexual intercourse and thinks that there's a possibility she could be pregnant. It can be taken within 72 hours of intercourse, not just the morning after. And the pill works because it has a synthetic progesterone-like hormone, and if it's taken early enough, it reduces the chances of a sperm reaching a fertilized egg. But in most cases, it prevents pregnancy by stopping a fertilized embryo from implanting into the uterus. So plants, like animals, have communication systems that allow coordination between different parts of their bodies, and they too must respond to changes in their external and internal environments, like we do, like we learned in Chapter 14, homeostasis. Most plants involve changing some aspect of their growth to respond to factors such as gravity or light or water availability. But they can also respond fairly quickly to changes in carbon dioxide concentration, lack of water, grazing animals, or infections from fungus and bacteria. Some of them are brought about by quick changes in turgidity, as happens when stomata respond to changes in humidity. But so let's first talk about electrical communication in plants. So plant cells have electrochemical gradients across their cell surface membranes in the same way as in animal cells. And they also have resting potentials. As in animals, plant action potentials are triggered when the membrane is depolarized. And in at least some species, some responses to stimuli are coordinated by action potentials. The sensitive plant, mimosa, responds to touch by folding up its leaves, right? So microelectrodes inserted into leaf cells detect changes in potential difference that are similar to action potentials in animals. And the depolarization results not from the influx of positively charged ions, but from the outflow of negatively charged ions. Repolarization is achieved in the same way, but it's similar to those transmitted along the neurons of animals. The action potentials travel along the cell membranes of plant cells from cell to cell through the plasmodesmata and are lined by the cell membrane. So the action potentials generally last much longer and travel more slowly than they do in animals. There are a lot of different stimuli that trigger action potentials in plants. Chemicals coming in contact with the plant's surface can trigger them. Okay? The Venus flytrap is a carnivorous plant that obtains a supply of nitrogen by trapping and digesting animals like insects. Okay? The specialized leaf is divided into two lobes, and the inside of each lobe is often red and has nectar-secreting glands around the edge to attract insects. Each lobe has 3 stiff sensory hairs that respond to being deflected. The outer edges of the lobes have stiff hairs that interlock to trap the insects. And the surface has many glands that secrete enzymes for the digestion of trapped insects. The touch of a fly or other insect on the sensory hairs of the inside of the folded leaves of the Venus flytrap stimulates action potentials that travel very fast across leaf, causing it to fold over the trap and trap the insect. So chemical communication in plants. Chemicals are known as plant hormones or plant growth regulators, and they're responsible for most communication within plants. But unlike animal hormones, plant growth regulators are not produced in specialized cells within glands, but in a variety of tissues. They move in the plant, either directly from cell to cell or are carried in the phloem. Some may not move far from their site of synthesis and may have their effects on nearby cells. So there are auxins, which influence many aspects of growth, including elongation, which determines the overall length of roots and shoots. And there are gibberellins, which are involved in seed germination and controlling stem elongation. So abscisic acid, or ABA, is another plant hormone which controls the response of plants to environmental stressors, such as shortage of water. So plants make several chemicals known as auxins, of which the principal one is IAA. Here we refer to it as auxin in the similar in the singular. Auxin is synthesized in the growing tips of shoots and roots where the cells are dividing, and it's transported back to the shoot or up the root by active transport from cell to cell, and also to a lesser extent in the phloem sap. Growth in plants in this way occurs at the meristems, such as those at the shoot tips and root tips. Growth occurs in 3 stages: cell division by mitosis, cell elongation by absorption of water and cell differentiation, and then auxin is involved in controlling growth by elongation. So the gibberellins are plant growth regulators that are synthesized in most parts of the plants. They are present in especially high concentration in young leaves and seeds, and are also found in stems where they have an important role in determining their growth. So the height of some plants is partly controlled by their genes. For example, tallness in pea plants is affected by a gene with 2 alleles. If the dominant allele is present, the plants can grow tall, but plants homozygous for the recessive allele will remain short. The dominant allele of this gene regulates the synthesis of the last enzyme in a pathway that produces an active form of gibberellin. Active gibberellin stimulates cell division and cell elongation in the stem, causes the plant to grow tall. A substitution mutation in this gene gives rise to a change from alanine to threonine in the primary structure of the enzyme near its active site, producing a non-functional enzyme. So this mutation has given rise to the recessive allele. Homozygous plants are genetically dwarf as they do not have that active form of gibberellin. Applying active gibberellin to plants, which would normally remain short, such as cabbages, can stimulate them to grow taller. Okay, so that is it for this unit. Okay, lots of details, lots of memorizations. Really, really important to understand about action potentials and movement along neurons, activity and axons. So make sure that you're really nailing down all those details, and I will see you in class. Thanks.